The sea spider lives in the Antarctic, Arctic, Caribbean, and Mediterranean seas and feeds on hydroids and bryozoans. They exhibit a characteristic known as polar gigantism, whereby they are generally larger than family members in other parts of the world, as they live close to the poles. These organisms lack a respiratory system in the body, and most of the organs are found in the legs. The diffusion of gases through the thin legs allows respiration to take place. It was initially believed that some of the ATP generated by mitochondria in the respiratory chain may be converted into heat. However, through the study of the mitochondrial genome of the sea spider by Antonio Carapelli et al., it was found that there was no specific gene that coded for this characteristic that would be considered as an adaptation to the low temperatures.
Megathera novangeli, otherwise known as the humpback whale, can be found living in cold environments such as in the Antarctic Ocean. They primarily feed on Antarctic krill, an extremophile crustacean found in these waters. An adaptation to the cold involves retaining a layer of blubber made of fat trapped in a mesh full of collagen to provide insulation for these organisms and to provide fat reserves to sustain high energy in long-term migration. They have a unique way of feeding, which involves herding their prey and coordination with other whales of their species. This is called bubble netting. The whales swim in a circular motion up and down their prey, blowing bubbles to create a whirlpool in which other whales can swim through and feed. A research paper carried out by the NOAA, National Ocean Service, recorded 300 bubble net feeding events from 9 humpback whales. 2 kinematic techniques observed to be the most efficient were upward spirals and double loops.
To avoid predation in the first place, krill hide by spending their days dwelling near the ocean floor. They only rise as night falls to feed off the phytoplankton on the undersurface of the Antarctic ice. Krill then mechanically rake the algae using their thoracic maxilliped appendages to act as a filtering system, preventing the escape of food as small as 1 micrometer. Interestingly, Antarctic krill have bioluminescent organs, which emit a yellow-green light in 2 to 3 second periods. These highly evolved organs contain a lens at the front and a concave reflector at the back, and their rotation is dictated by muscular relaxation and contraction. A University of Gothenburg study went on to find that the relaxation of the krill causes luminescence, as the relaxation of the thick and thin filaments surrounding the capillaries enable them to broaden and drain the hemolymph into the organs, creating luminescence. Some believe this highly evolved torchlight mechanism is used to erase their shadow from the hungry eyes of their predators, whilst others suggest it is a communicative method used in the dark of night.
The Antarctic icefish have certain proteins in their cells that prevent their body freezing, called antifreeze glycoproteins. They are structures called glycopeptide repeating units and work by causing a process called thermal hysteresis, which prevent ice crystals from forming. As their cells are made up of mostly cytoplasm, and the cytoplasm is 70% water, ice formation would expand them so much that they would burst, eventually causing death. By lowering the freezing point of water to around minus 10 degrees Celsius, also known as supercooling, thermal hysteresis allows water to remain as water, well below freezing point. The chemical structure of the antifreeze glycoproteins allows them to attach to the ice, preventing other molecules to grow here, and more proteins can attach. This means that there must be more cooling in order to maintain ice growth, along with protein growth.